Hi there, and welcome back to our second topic about the origin of life. Previously, we talked about stromatolites, structures potentially produced by microorganisms. But this video focuses on direct evidence for life in the form of the microscopic fossils of the organisms themselves. So just a quick recap of the background or the setting for today's topic. So the Earth formed a bit over 4.5 billion years ago, but the first 500 to 700 million years remain pretty mysterious because of limited evidence. The oldest known sedimentary rocks are around 3.7 to 3.8 billion years ago, although they've been extensively heated and deformed. But these rocks, in Greenland in particular, contain structures that have been interpreted as stromatolites by some scientists, but that are still very controversial. So the oldest well-preserved sedimentary rocks date from about 1 billion years after the formation of the Earth, about 3.5 billion years ago. And that's what we're considering today. So this takes us to a remote part of Western Australia, to a 3.46 billion year old unit of rocks named the apex chert. So chert is just a type of rock. This region of Western Australia also contains stromatolites of a similar age that are pretty widely accepted as structures formed by bacteria. But today we're considering something that's been much more controversial. So in 1993, paleontologist Bill Schopf described microscopic structures from the apex chert as fossils of bacteria. And he identified about a dozen different types, some of which are shown in these photos here. They're all really small, smaller than the width of a single human hair. This would be a tremendously important discovery, the oldest, really strong evidence for life. But first, some background on bacteria and the classification of life to give you some info on that. So all life on Earth evolved from a single origin called the last universal common ancestor. And evolutionary relationships are often shown with these kinds of branching trees, like the ones here, where the tips of branches at the top indicate a particular organism, and the branching points are where two organisms split from a common ancestor. This one is just a schematic. There's obviously a lot more than 4 or 5 organisms in each of these big groups. But life is classified into 3 main domains: bacteria, Archaea, or sometimes called Archaebacteria, and Eukarya, or Eukaryota. You'll learn about the evolution of eukaryotes later on, but they basically evolved when an Archaea decided to take on a bacteria as a partner. And eukaryotes like us have those bacteria partners still today. So you've probably heard about bacteria before. At the most general level, they're organisms that only have a single cell, although they can live in groups of multiple cells. Their cells are fairly simple, basically just a container for DNA and some other things. And we most often know or hear about bacteria because some of them cause diseases, but bacteria live everywhere in the ocean and in lakes, in soils and in rocks, and even miles down inside the surface of the Earth. They're extremely important in all ecosystems. For example, some bacteria produce oxygen through the process of photosynthesis. So when you think of fossils, you probably think of dinosaur bones or shark teeth or clam shells, so it might be surprising that bacteria can become fossils. Well, it's extremely rare, yes, but it can happen under special circumstances. And the photos on the right here show bacteria fossils that are around 800 million years old and are widely accepted as actual fossils of bacteria. And although they have more detail and are better preserved, the overall appearance is pretty similar to the apex chert specimens on the left. Well, in fact, the apex chert specimens were originally compared with a specific type of bacteria called a cyanobacteria. And this comparison was made on the basis of size primarily, and that's shown in the graph on the left. So most bacteria have very, very small cell sizes, only a few microns or micrometers in size. So that's the tall peak in the dotted line on the left side of the graph, showing that most bacteria have cell widths of, say, only 1 micrometer or micron. But living cyanobacteria, or particular types of them at least, tend to have larger sizes and also a wider range of sizes. That's the broad peak in the thin line from about 3 to 12 micrometers there. The 11 species of the apex chert filaments have a similar range of sizes, and that was the basis for this argument that they are cyanobacteria. But this identification would be even a more significant finding because cyanobacteria use sunlight for energy to split water molecules for energy, and they produce oxygen at the same time. This is a process called photosynthesis. Well, you'll learn more about this a bit later on, but oxygen didn't first accumulate in the Earth's atmosphere for another billion years after these specimens. So if these really are cyanobacteria that produce oxygen, that would cause quite a puzzle. But actually, the specimens didn't really attract much scrutiny until a decade or so after their initial description. But let's think about some ways to test the hypothesis that these structures are bacteria, or even that they're cyanobacteria for that matter. What would we expect if they are microfossils? And therefore, what additional questions could we ask? Or what additional information could we collect to make it more convincing, or even to contradict the idea? Think about it to yourself for a moment if you want. You can even pause the video to think about some questions. So here's 3 things that we'll consider in the next video. First, are the specimens found in rocks that formed as sediments on the seafloor? Yes, I mean, it's true, bacteria can live in almost any place, but cyanobacteria need sunlight for photosynthesis, so they live in shallow water in the ocean or in lakes. Second, are the structures actually made of cells, like you would expect in bacteria? And finally, are they made of organic carbon compounds? What is the material that makes them up? Well, thanks for listening, and make sure to tune in to the next video to hear about the debate over these questions.